pleasure to introduce Dr. David Lipson from San Diego State University. Dave, much like Forrest, shares a bit of history with Mike Graham, and that you're both at Santa Barbara City College at the same time.
Oh,
And from there, Dave went on to get his degree from Berkeley. He then went on to get his PhD and do a postdoc at the University of Colorado at Boulder. Dave was on the faculty with me down at San Diego, and he and I have had a chance to teach classes together in ecology and recently in clinical physiology. It's been a lot of fun doing that. And Dave, as you'll see, one thing I like about Dave is his kind of approach to things, the way he describes things. He kind of has these other interests, and one of them is, as we've talked a lot about, is music and building instruments. Dave has a house full of very interesting instruments where he takes apart guitars and builds huge things with pickups and all these things, and he creates his own instruments and then tries to play them. Only a little bit. Yeah. Dave has had the opportunity to work in a wide range of ecosystems, and as you'll see, he's one of the things that really excites me is his work kind of near Barrow, Alaska. It's one of the really things I find particularly cool. I really love the Alaskan landscape, and I think today he's going to be talking to you a bit about that today. So without further ado, I'm going to introduce Dr. David Lipson.
Thanks a lot for inviting me up here. This is such a beautiful place. It's a great excuse for me to come up here and hang out a little bit. So yeah, as I think I'm turning into an eccentric old professor already, because more and more silly poetry is working its way into my science, and I'm hearing it from the reviewers constantly. I'm still getting smacked down, but I guess I'm just caring less and less. But so this is an opportunity where I can get away with a silly title like the Cornucopia of Anaerobia. But I'm going to explain where this title comes from. And basically, there is working in this wet, squishy environment up in the Arctic coastal plain, and I've encountered a really stunning variety of anaerobic pathways, things I had never actually heard about when I had started working there. So the Arctic coastal plain near Barrow, Alaska, it's about 71 degrees north, our northernmost city in the United States. So the landscape is mostly wet and anoxic, suboxic. So there's a permafrost that prevents drainage, and the sphagnum mosses are extremely good at soaking up water. And so it is wet and squishy. And so the structure of my talk, I'm kind of going to weave together 4 separate studies that are all related. And sort of tell you what I've been doing for the last 8 or 9 years up there in Alaska. So various anaerobic pathways. First of all, a very general study that I've done most recently, looking at how oxidation reduction potential in these soils structures the microbial community, and how that really is the driving force of the microbial community. I'm going to talk a lot about iron 3 and humic substances as electron acceptors. So this ecosystem is dominated by anaerobic respiration, and those seem to be, particularly iron, ferric iron, the dominant electron acceptor. And how these electron acceptors can compete with methane by providing a more thermodynamically favorable pathway. And then if I have time, I'm going to talk about what I'm getting into most recently. Iron is totally 5 minutes ago, so now I'm going to be talking about chlorine cycling, which is another thing that I had no idea was really even a biological cycle. So first of all, explaining the Cornucopia of Anaerobia, this is a close-up of a wet, moss-dominated little patch of tundra. You can actually even see a little place where I've stuck a pH probe, probably right about there. Oh, I'm sorry, first I have this. Anyway, and so there's just to me, it was a really surprising variety of anaerobic pathways. So iron oxides, both in insoluble mineral form, like we find as rust, and also in soluble forms, they're solubilized by siderophores and organic matter. Humic substances, which are complex organic compounds in soils that give soils their dark color, and all they're also in aquatic systems, I imagine as well. There's a little bit of oxygen, there's a little bit of sulfur, but I've been focusing mainly on these things, and then more recently organohalide respiration, where microbes can actually use carbon-chlorine bonds to accept electrons, and that releases chloride. And originally only studied in terms of like dry cleaning solvent waste, but it turns out there's a natural chlorine cycle that happens in relatively pristine places. And then of course all this impacts methanogenesis, the production of methane, but also relatively recently people have discovered that methane can be oxidized anaerobically. And so some of these alternative electron acceptors might come into play in terms of not just competing for substrate in the production of methane versus CO2, but also oxidizing methane using anaerobic pathways. So some overview of the electron tower. What I generally teach in my microbial ecology classes is this helpful mnemonic, "Oh no, my feet's so cold." People heard that one? I've already messed it up with a couple of these other things. But in general, the most powerful oxidant would be oxygen, and then nitrate, manganese, iron, and then sulfate, and then at the bottom, CO2, which would produce methane. I'm adding in a couple things because we keep discovering new and bizarre alternatives. But taking a substrate which could also drive methanogenesis in the presence of all these other electron acceptors, you're more likely to get anaerobic respiration, CO2 production. Because, frankly, this is sort of this is the bottom of the barrel in terms of thermodynamics. Like, there's the smallest amount of energy available from producing methane compared to if you have an actual electron acceptor. So all of these things can compete. There could be organisms that can have these pathways which would compete for the acetate or hydrogen. And it also turns out some methanogens themselves are capable of switching their metabolism to this more energetically favorable pathway. So there are some methanogens that can, in the presence of humic substances, for example, would reduce the humic substances and make CO2 instead of methane. So this methane could leave the soil at that point, could get oxidized on the way up by the oxic layer on the top. And I should mention that some oxygen could probably sneak its way down to deeper layers of the soil through sedge roots, things like that. The oxygen we only see in the upper few centimeters of the soil. And more recently, people have discovered that there are these, not for absolutely every one of these electron acceptors has been demonstrated to be able to do this, but thermodynamically, they're all possible. The first reports of anaerobic oxidation of methane came from sulfates, seem to be the electron acceptor. I'll show you how, this is actually a consortium. This is not one group of microbes, but actually two carrying this out. More recently, they found a single microbe that can use nitrite actually to oxidize methane. It produces its own oxygen in the process and oxidizes methane. It's kind of amazing. So there's a rich assortment of pathways happening in this ecosystem. I'm going to focus on iron and humic substances and maybe a little bit of chlorine. We don't have a lot of sulfate in this system, even though it's right next to the ocean. You walk around this anaerobic landscape, you don't smell a lot of sulfide, so it's mostly bound up in organic matter, and so there might be a sulfur cycle, but it's kind of cryptic. And nitrate, in general, it's too anaerobic to see a lot of nitrate, except for in some of the higher raised areas of the landscape. Okay. So here we are in the northern tip of Alaska, about 71 degrees north. And you can see all these features. These are either thaw lakes that fill as the snow thaws in the summer, and are, again, drainage is blocked by permafrost. And over time, there's some erosion, and some of these drain. So some of these are filled with water and are currently lakes, and others used to be lakes, and those are called drain-thaw lake basins. They then vegetate over time, and I'll show you later on in this talk that people have done a nice job, as you can see 14 to date, how long ago they were lakes. And so there's actually soil development. So it's a nice chronosequence. We have very young ones with less than 50 years, very small amounts of organic matter accumulation, to ancient ones that are 5,000 years and have very thick organic layers. So, it's a convenient chronosequence to use in soil development. If you zoom in a little bit closer, you can see these ice wedge polygons. These older basins, the old and ancient, the things that are about thousands of years, hundreds of thousands of years, start getting these ice wedge polygons. So the water tones, they coalesce, form ice wedge polygons that lift up certain areas of soil, making water cool in other areas, and then that can cycle. And so these ice wedge polygons create these redox gradients where you can have a relatively aerobic area that's sticking up by maybe half a meter relative to the bottom of the polygon a few meters away. And so there are these, again, it's mostly anaerobic, but there are these strong gradients in oxidation reduction potential, redox. And so this first study I want to show you is very descriptive, just looking at how the microbial community varies with depth and with microtopography across this landscape. So zooming in, here are some ice wedge polygons. These are probably about 30 meters in diameter on average. And here it is on the ground. The rims of these low-centered polygons are nice places to eat lunch after picnics. And in the ancient, these very old terrain thaw lake basins, you can actually get ponds forming there, and so you can have fairly deep, not just ice wedge polygons, but actually little ponds within these lake areas. So we would stratify our sampling from these rims of low-centered polygons to the centers of low-centered polygons, where it would be wet and anoxic and slightly higher and drier. Oh, and here's a picture of Ted and Archaeal using a zipper cord to get it. Everyone loves to pose with that thing, big gas power drill. So I want to acknowledge the Earth Microbiome Project, which I think is now defunct, but Janet Jansen, who was at Lawrence Berkeley Labs, who's now at Pacific Northwest, basically had many of my soil sampling gave me over 2 million sequences, so I'm still suffering future shock from this, that I can have 2 million sequences. When I was in grad school, I had 200, each one I would open like a Christmas present. So now I have 2 million. It's all automated. But this is a very ambitious project where they were doing high throughput sequencing of 16S ribosomal RNA genes to describe microbial communities around the planet. And so we had a depth profile in high and low areas of the landscape. And just to give you a basic idea, this is a principal components plot showing you there's a metric that's very convenient called the unifrac. You can compare entire communities, and unifrac provides a metric for comparing entire microbial communities between two samples. And using the unifrac metric, you can see the clustering of, so the color shows you depth. So 6 centimeters are here in the light green, 16 centimeters in blue, and then deeper, the red and pink. And the shapes, triangles versus circles, show the topography. So R means a rim, so that's a high area. C means a center, a lower wet area. And so you can see the depth has a very clear influence. You can see the shallow stuff separating out quite a bit. And you can see near the surface a bit of the effect of topography in that these high areas of the next layer down are clustering with the shallow soils, right? So you kind of have the same gradients, but they're maybe offset by a few centimeters because the ice wedge is pushing up the rims. And then once you get to a certain point, 26, 36 centimeters, the topography is really hard to distinguish. At that point, it's all probably pretty anoxic. One thing I thought was very interesting was that overall diversity declines with depth and with microtopography. So here you see the estimated operational taxonomic units. I don't know if there's a lot of microbiologists who deal with sequencing stuff, but because defining species, especially with just sequence information, is problematic, we tend to just settle for things called operational taxonomic limits, which are defined as some arbitrary similarity. This was 97% similarity equals 1 operational taxonomy. So we don't have to say the word species. But anyway, again, this is my first time I got to play with 2 million sequences. So these rarefication curves are kind of a lot further along the way that I've ever gone. And they still haven't saturated. It's kind of cool, meaning that just with brute force, you might never fully sequence a soil. That was kind of something that dawned on me. You probably have to really go after the rare things. There's actually a famous, or I think like a 1961 Preston paper about how at a certain point with random sampling you're only going to get like 80% of what's out there anyway. He wasn't talking about sequence data from microbes. Seems to hold true. But anyway, I was struck by both. So this is the topography. So higher and drier, wet and more anaerobic, and then with depth. And these, by the way, are extrapolated. I have fewer sequences from the deep areas, but still it's quite a significant trend. So this is extrapolated with a sort of a bootstrapping approach. But you can see from 6 centimeters to 16, there's a pretty steep drop. It seems to continue throughout the depth. And so that, and I should remind you again that in most parts of the landscape, most of the oxygen is actually already gone by 6 centimeters. So it's definitely getting less diverse as it gets more anoxic, but there's still a big difference from having maybe iron around as an electron acceptor to having maybe no electron acceptors whatsoever. So I don't expect you to be able to read this table. I have, gazillions of tables like this where this is showing correlation coefficients of, this is my proxy for redox. I took the soil, extracted an acid, and looked at the relative oxidation reduction state of the iron in the acid extracts. And the point here is just that all these microbial groups, in general, the best predictor was this redox variable, better than pH or organic matter content. I mentioned pH and organic matter because they're far more frequently mentioned in the literature in soils about describing microbial communities. Redox doesn't get a lot of attention. Of course, admittedly, I'm working in a much more anoxic system than most people. And regressing the principal components that I showed you earlier from this unifrac analysis that describes the overall community. Again, this redox variable, iron 2 over total, so this is the higher that variable is, the more reducing the system is. That was a very powerful variable, better than anything else. And then there were some second order effects in dissolved organic carbon, organic acids, and specifically this organic acid lactate was maybe explaining some of the variation in the community after the redox had eaten up most of the error. So there were some subtleties in there, but redox really was defining the community, both in terms of who's there and in terms of diversity. So redox is clearly driving this system. Let me, and again, I told you I'm sort of becoming an old fart, getting more philosophical. So I had to, the reviewers are making me take this quote out of this paper. I really love it. So Albert Georgi said, life is an electron looking for a place to rest. And apparently he also said, what drives life is the little electric current. So, yes, redox is important. I've seen some other papers that picked up this theme and relate this to something called diversity energy theory. Basically, more energy should lead to more diversity. But in soil work, usually this is related to electron donors, how much sugar there is in a system, not how much energy is available. These are organic rich systems, a lot of organic matter accumulating. So what might actually dictate the energy availability is the other side of the equation, the oxidants. So oxygen, terminal electron acceptors. Okay. So this is a bit of an exaggeration, but down at the depths, the diversity was declining to a point where it was mostly fermenters. So Clostridium bacteroides, which are incidentally the same things that are in human guts, by the way. These are not the same species, these are living at 0 degrees C. Probably wouldn't be happy in our gut, but the same kind of metabolism is going on there. And so I was a little bit surprised to see that probably a lot of the microbial diversity that boggles the mind are actually carrying out, a lot of them are probably aerobic, but certainly a lot of these things are at least carrying out anaerobic respiration using subterminal electron acceptor. And the fermenters are not as diverse as a group. At least it's a restricted subset. So moving on, this is actually an older study, but that is a really nice general introduction to the system and the microbial community there. We also have metagenomic data, which I previously showed you was only 16S data, 2 million sequences of ribosomal DNA that just show you the taxonomy entries there. Here we can get at more functional genes by doing the shotgun sequencing. And so this shows you a cross-section or a typical soil core. There's, depending on where you are, this is probably from an old basin where we have a reasonably thick organic layer. You can see a mineral layer and a little ice lens down there, actually. The active layer is what thaws at the end of the summer. It's about 30 to 35 centimeters in the system. Below that, permanently frozen. So yeah, you can see about 30 centimeters, if that's clear. It's sort of the boundary between the active layer and the permafrost. So we broke up these cores into 10 centimeter chunks and extracted DNA and just sequenced a decent number of sequences. Just to get an idea of what the community looked like in terms of anaerobic metabolism. So first of all, when you put together all the genes and look at their closest hits in the database, the two most abundant genera end up being Solobacter and Geobacter. Geobacter is a famous iron-reducing bacterium, so that wasn't surprising, because at this point I already knew that iron reduction was very important in the system. Solobacter is part of this Acidobacterium phylum, which is all over soils, but very rarely cultured. We have this problem that there's a small percentage of things in the environment you can actually culture in the lab. And Acidobacteria, we're actually getting a little bit better at culturing them. You just have to be patient. Wait for 3 months, literally put tape around your plate so it doesn't evaporate. And you might get some Acidobacteria colonies that are just tiny. And again, it comes out in the database as a single genus, but the database doesn't have nearly enough genome sequence to maybe really know. This could actually represent a cluster of uncultured things, and the closest culture relative happens to be this one. Actually, it's not even a culture relative. It's something that was pieced together from metagenomes that they could tell was a single one. So this was just a little bit more information about the Acidobacteria. This is an old reference, but it shows you the basic, it still shows you the basic predicament of soil microbiology, in that this is the cultivated, relative proportion of cultivated microbes from these different phyla, and these are the ones that were just shown from environmental surveys. And you can see there are these groups that are just have a handful of cultured representatives, and the rest are just sequences in the environment. So we don't know much about Acidobacterium. There's a handful of cultured things, but they're probably very physiologically diverse. And we know at least one of them is an iron reducer. So just looking at the relative proportions of genes as we go down the profile, we can see these are respiratory reductases. So the anaerobic ones increase with depth. Cytochrome C is an aerobic one that declines slightly with depth. And this terminal cytochrome D is one that's found under microaerobic conditions. Doesn't seem to show much of a trend. These are methanogenesis genes that are certainly lowest near the surface, which makes sense. These guys are strict anaerobes and should be inhibited by oxygen. Acetogenesis, these genes are pretty low. There's this idea out there that under acidic conditions, acetogens should outcompete methanogens. These are soils of pH 5 or so, but that's not the case anyway. So it seems like there's more methanogenic genes than acetogenic. And just looking at pathways of known organisms that carry out these various anaerobic pathways. So these are iron-reducing bacteria. These are just the known ones. Geobacter, for example, this genus that very widely studies iron reducers. I haven't included all the other things that might be doing it, but this is just an index saying, okay, these are known iron reducers, these are strict fermenters, Syntrophic, these are dehalorespirators. Those are the ones that are using the carbon chlorine. And so anyway, you can see sulfate reduction, and yeah, syntrophic. Those guys are fermenters that work in cahoots with pathogens and other vectors. So many anaerobic pathways, iron reduction certainly seems to be a popular one. But the hard thing is that all this type of analysis is limited by what genomes have been sequenced and published and how well annotated they've been. So for iron reduction, not nearly enough is known. It seems to be really widespread throughout the microbial world, but it's only been studied in a relatively small number. So whereas these other systems, denitrification, sulfate reduction, arsenate, DMSO, methyl ammonium something, all these different pathways have more or less all the genes in the pathway worked out. For iron reduction, a lot of it is still a work in progress. So I chose these Decaheme Cytochromes. I looked into that because, and I'll show you why, as a indicator of iron reduction in the system. So these are interesting molecules. Why would it, so we have cytochromes in our system, and we have obviously a heme group in our blood, but we only have one usually in our proteins. Why would a protein need 10 heme groups, right? But each one of these little red dots is a heme group. So this really seems to be about long distance electron transport. So this is inner membrane and outer membrane, plasmic space of a gram-negative microbe. There's all these multi-heme cytochromes that seem to be transporting electrons out into these big extracellular electron receptors. These guys can't breathe in iron like we take in oxygen, right? They have to essentially have ray guns on their outer membranes and shoot out the electrons, right? So, and that's why these same organisms can be used to power electrodes. If you see microbial fuel cells, these are the things that in nature they're using iron oxides or complex humic substances in soils that they can put in microbial fuel cells and generate a little current. Because they have the ability to shoot those electrons to an electron acceptor outside the cell. So I put a little map, a tree together of the hits in my metagenome to known Decaheme cytochromes. And the nice thing about this is there's motifs, like every heme that gets bound has a specific amino acid sequence, so it's really easy to find. And then there's also a iron oxide binding motif that I have those labeled in orange. So I'm pretty confident that these things are not just Decaheme cytochromes, but are actually binding iron oxides. And the interesting thing about it, there's a whole bunch of well-known Geobacter, Anaerobicobacter, Rotiferix, these are, and Schwannella, those are the classic iron reducers that are known. But these Stolobacter, Pteroglobus, these are the Acidobacteria group. And so it really looks like this big, mysterious, uncultured group is contributing to iron reduction in this soil. So I was very excited to see that. And again, that was the other most common genus in this soil. So more evidence of iron reduction really ruling this system. And by the way, it keeps going up and up. I have an isolate that has, this one was found both in the metagenome and also I have a dye culture. I had it before it died. 19 hemes. That's got to be moving some electrons a really long way. 19 hemes to work with. Okay. So again, the metagenomic data really show a system that's filled with anaerobic pathways. And these Decaheme cytochromes seems to be a good indicator for iron reduction, and Acidobacteria seems to be important in that process. Now I want to put this in a global context. This is a polar view of the planet and mapped with amounts of organic carbon in the soil. And it's always a little bit hard. The Aleutian Islands are the easiest thing to see because they get disoriented. So yeah, that must be right there. Barrow is that point above the, it must be the Arctic Circle. Purple is the highest amount of carbon. So there's, just in the last few years, a really large correction in the global estimates of soil carbon in permafrost and permafrost affected areas. And so now the estimates are around 1,700 petagrams which basically that was the amount, close to the estimate of the total global soil carbon. So lots of carbon in these northern systems. And of course, climate change in the Arctic has already been observed to be much faster than the rest of the planet. And these are the most recent scenarios. This RCP 2.6 is like extremely optimistic. Like if we got together like right now and started limiting carbon, we could maybe approach this goal. This is the more business as usual aggressive fossil fuel scenario. And you can see this polar amplification in the darkest colors in both models, or both projections, you see in the Arctic. And so there's various kinds of feedbacks, loss of sea ice, absorbing more heat, etc. So the Arctic is really taking it in terms of climate change. And of course, people are concerned about positive feedback. Carbon stored in permafrost, warming being released, amplifying CO2 and methane production. So like I already told you about, in this case, we have an anoxic environment where drainage is blocked by permafrost. Iron reduction is a dominant process. One question for my area is, as that active layer gets deeper, as the system warms up, how will that alter the biogeochemistry of the system? So if there is, specifically, if there's more mineral iron in the deeper layers of soil, that could promote more iron reduction, and actually that could compete with methane production, which would in some tiny way be good news, I suppose. Although ultimately it all turns into CO2, and it really just sort of changes the short-term versus long-term forecast of climate change. Still, important questions. I don't want to be too negative about it. So again, here's the picture. My point was that the mineral layer, in some cases, the really ancient soils in particular, if this organic layer accumulates to above 30 centimeters or so, this stuff gets pushed down to the permafrost. And so that's an interesting aspect of this area where I work, is the organic layers, and some of them, because they've only been accumulating for, in some cases, less than 50 years, there are mineral layers that are still in the active layer. And so that might be, maybe the system working in this kind of freakish, and that's why people were surprised when I said iron reduction was a big deal in the Arctic. But if the permafrost, if the active layer gets deeper, the permafrost starts to melt, more stuff in the mineral layer should in theory become active or available for cycling throughout the profile. So how would that affect carbon cycle in the Arctic? By the way, oh yeah, I think you can see in this light. Do you see that it's actually iron buried deep down there in the core? You can see the orange rust color? So, again, this is pretty cool because that's a deep anaerobic part of this is down to 30 centimeters deep. I'm still working out how this stuff gets reoxidized every week because the water tadpole doesn't even usually get to 10 centimeters after a long dry summer. So it's still a little bit unclear how the iron oxide forms after it gets reduced, but still working on that. But like I said, if iron comes from the buried mineral layer, which is getting progressively buried deeper and deeper as organic matter accumulates, what will happen is the permafrost recedes it. So again, here's a picture of these drained thaw lake basins, and here is overlaid some work by Hinkle and Decaheme and others that use C14 ages and morphometric stuff, so remote sensing, a variety of methods to age these different basins into young is 50 years in yellow, purple are the medium, which are up to 300 years, red are old, which is 300 to 2,000, and then the green are the ancient, which are up to 5,500 years old. And over time, organic matter accumulates. This is a dominant form of the Arctic coastal plain. The landscape's over 50% are these thaw lake basins. And another thing that happens as they develop is you have polygonization. The ice wedges build up, and so you have more topographic, microtopographic variability for the landscape. So ice wedge polygons, you have the young ones are very flat and just all squishy, whereas there's nice places to eat lunch in the ancient basins, like they should be. So I did a very straightforward study where I just removed iron from different depths. Here they're all summed together. Dropped them in acid so the iron would be oxidized abiotically. Took them to the lab and just did a survey of the amount of iron 3 versus iron 2 over the season. And towards the last half of the growing season, the iron 3 was dropping, meaning it was all being reduced in these 4 different age basins. Doing some basic back-of-the-envelope calculations of how much iron got reduced over what time period, and looking at how much CO2 is coming out of that landscape, I came up with these sort of back-of-the-envelope calculations of how much was iron reduction accounting for ecosystem restoration. And pretty impressive numbers, so 40 to 60%-ish of ecosystem restoration being accounted for by the reduction of iron. There's all sorts of caveats I could go into, but still, it was quite impressive. Large amount of iron being reduced, and that can account for a lot of the CO2 that's going out of the soil. The next question was, how does iron availability? Oh, and I should point out, that in the medium and ancient basins, it appeared to be possibly a little bit lower. So I want to look in a more replicated way. This is just a single, each one of these was a single basin. Went back the next year, did a more extensive survey of the landscape, and looked at how iron availability changed with organic matter depth, the organic layer. And what I have here are 2 proxies for how thick that organic layer is. Just the total organic matter content of the active layer and the bulk density. So a high organic matter content means a thick organic layer. A high bulk density means a lot of mineral layer, so a shallow organic layer, these high bulk density soils. So that's why you have a negative relationship with extractable iron going down as the organic layer gets thick, or in this case, these are shallow organic layers with a lot of mineral material. So it was like I expected. Soils with a lot of mineral layer in the active layer had more iron. So then the question is, we know it has a big impact on CO2. What is it doing to, I'll skip that. What is it doing to methane? So first of all, a really interesting metric to look at is the ratio of CO2 to methane that comes out of the environment. In theory, if you had a completely methanogenic system, so a bunch of organic matter, no oxygen, no nitrogen, no manganese, no sulfate, no iron, no humic substances, nothing to breathe, just hydrogen, CO2, acetate, say, you would get a 1-to-1 CO2 to methane ratio in theory. And the numbers that we're getting, so these are the amounts just dissolved in soil pore water at 2 different depths, or what came out of soil incubations in the lab. They're all much higher than 1. Some of them are in the thousands. Most of them are in the hundreds. So a high CO2 to methane ratio in an anaerobic environment really implies there's a lot of terminal electron acceptors, alternative terminal electron acceptors. And there's a bunch of gradients here. You can see this was in a flooding experiment. We actually put in dams in a big basin and raised the water table in part of it. That's a whole other story. So we but anyway, no matter where we were on the landscape, there tended to be very high CO2 to methane ratios. So that already is a sign that electron acceptors are available and interfering with methane production. And just, if you describe variations across the landscape, we see decreasing dissolved iron in areas with more available iron to eat. That's essentially decreasing methane in soil pore water with increasing iron. And the same trend with increasing organic layer thickness, more methane. So these would be more like ancient soils, not as much iron because the iron is buried deep under the permafrost, more methane there. And my student Kim Miller and a few other collaborators did a manipulation study where they had these paired cores. They either injected a soluble form of iron 3. It's iron 3 nitrilotriacetic acid. It's a commonly used one in microbiology experiments. It's basically got a chelated form of iron that will stay in solution, so you can work with it pretty easily. Or humic acids, which again in this case were just store-bought aldrich humic acids. So adding electron acceptors, then looking at the CO2 and methane flux. She had to do this experiment about 3 times before she finally found the right experimental design, by the way, because a lot of spatial variability across this landscape. Again, you can see these rims and low centers in the polygons. And so finally figured out that she was able to control for this variability by having a control right next to the treatment and subtract that. We could actually finally see an effect. So that was what's happening here. This is the difference between the control and the methane flux. This is before addition, and then after addition, saw a significant depression in both the iron and humic acid treatments. And that finally just got accepted. It's probably available online in some form. So iron 3 and humic acids do seem to suppress methane fluxes. How's that working? It doesn't seem to be as simple as just competition. This is some other metagenomic data comparing 4 basins of different age. These are the amount of methanogens, the relative proportion of methanogens as we go from young to ancient. Here's where we actually have the smallest amount of iron and the highest amount of dissolved methane, but actually a relatively small number of methanogens. So it's not that the methanogens are being displaced from the system. It could, however, be that the methane is being produced and then oxidized when you add more electron acceptors. So Kim did these pool dilution experiments where you have soil in jars, the high methane headspace, and it's labeled with C13. And by looking at the changes in the total amount of methane and the C13 label, you can see simultaneously the production of methane and the consumption of methane at the same time. So it's kind of a nice method. It's a little bit expensive and a pain, but you can get a lot of information out of it. At the same time, she added a bunch of different potential electron acceptors to see if that would influence the system. And it turned out that, so yes, first of all, yes, anaerobic oxidation of methane was occurring. And these are under ideal conditions, so absolutely jam-packed levels of methane in these jars. Under those conditions, the methane was actually in steady state, so as much as was being produced at any one moment was being consumed anaerobically. That was a pretty unrealistic methane concentration, but if you get pretty far down in the soil, methane goes up from parts per million to maybe a couple percent, so it's not totally ridiculous. But again, these are really optimal conditions. And the iron 3 treatment, at least at some depths, did seem to increase the decay constant for methane. So there is some evidence from her study here that iron 3 might be serving as an electron acceptor for anaerobic methane oxidation. This is one of the first papers I saw talking about anaerobic methane oxidation. Now they call it AOM to differentiate it from ammonium ionoxygenase if there's any gene doctors out there. Anyway, it's usually a consortia. These are archaea carrying out methanogenesis backwards. Instead of taking hydrogen, CO2, or acetate and making methane, these are in areas with really high levels of methane. Methane is getting stripped of its electrons, producing these end products, and then sulfate-producing bacteria are using up the hydrogen and acetate to make that all thermodynamically plausible. Here's a picture of the archaea labeled in red and the sulfate-reducing bacteria around them. So, really cool paper. So, something like that might be happening in our system only with iron-reducing bacteria instead of sulfate-reducing bacteria. Okay. And when we saw this, I did decide to go back into our databases and look for genes that were related to organisms that carried this out. So another awkward acronym, ANMEs are anaerobic methanotrophic urtiarchaeota. If you're falling asleep, go ahead and try to say that with me. Urtiarchaeota. The ones in bold are sequences from our, MCRA. That's the gene that It's some kind of methyl something coenzyme reductase, coenzyme Q reductase, I'm sorry, I forget. But McrA produces methane, but also these close relatives of methanogens that are consuming methane seem to use that same gene. And those are the ANME 2A, 2C groups 3, 2A, 1. So you can see them labeled on this tree. And so we have some that are pretty closely related to this ANME 2 group. It seems suspicious. Did this with 16S as well, and have some sequences that are falling out close to this ANME 2 group as well. So it seems like there's a good chance the organisms are there. And the nice thing about having 2 million 16S sequences is you can find the rarest things there. Anything you're looking for, you'll find. This NC10 group is this methyl mirabilis oxifera, which is a beautiful name. These are the ones that use nitrite, they make their own oxygen, and use that to carry out methane oxidation. It's really surprising, just discovered in 2010 or so. These are pretty low numbers, but they are in the system, so. All right. So, iron reduction contributes significantly to ecosystem respiration, does decline with increasing organic layer thickness, although I was surprised to see a pretty large amount in the ancient soils. I didn't really point that out, but you have to remember that even the ancient soils were young ones, and that iron has been cycling and accumulating. So you have iron minerals forming in the organic matter. Now seems obvious to me, but at the time it was surprising. We have a few lines of evidence that iron 3 limits methane production, both observational and experimental evidence for that. And so, according to these data, you would expect that as the permafrost, and this is in continuous permafrost areas, where the permafrost shouldn't disappear altogether. I don't think, in any of the likely scenarios. So it'll stay as an anaerobic environment. There should be increased iron availability, and so maybe that would promote CO2 over methane production. Okay, so I don't know how full your brains are and how much time I have. What time do we start? Let me just tell you really quickly about what I'm doing next. I'm not going to explain every one of these arrows, but I am excited about the chlorine cycle in the Arctic. So again, I promised you a Cornucopia of Anaerobia, and this is sort of what I'm talking about. I didn't even know about this pathway when I started working in the Arctic. Chlorine has been considered to be generally inert, right? It comes down in rain as chloride. It's used as a tracer in hydrology experiments. It runs off. People studying biological transformations of chlorine were mostly interested in cleaning up like perchloroethane, like dry cleaning solvent spills and things like that. And there are, of course, some antibiotics that have chlorine on them. But there's mainly grouped in Europe, Sweden in particular, there's some interesting studies where they saw that, wow, actually it looks like there is in these relatively pristine forests a biological chlorine cycle. And so when we started seeing sequences like Dihalococoides, what's that doing in our soil? That's something that's been associated with, again, these like chlorinated solvent degradation. Started investigating the idea that there might be this biological chlorine cycle. So I don't want to necessarily go through all these cycles, but this is organic chlorine, chlor. And so this step here, going to liberating chloride, that would be dehalorespiration, or what's apparently the cool kids now call it organohalide respiration. We think that's happening. This can also happen photoheterotrophically by certain organisms. And this is also known to interact with the iron cycle, which is an interesting thing. So abiotically, iron, there's these Fenton type reactions that can make free radicals that make this hypochlorous acid, which can react with DOC. And this is one of those things that makes volatile organic halogens, things that are important in atmospheric chemistry. And so not only is the chlorine cycle interesting to be just for its own sake, but it interacts with the iron cycle in ways that could maybe even help regenerate some of our iron 3. I'm curious how that's happening. So just a little bit of preliminary data to show you that this seems to be happening in our system. Survey of dihalococoides. They are found predominantly in the anaerobic parts of the landscape. They seem to peak at the 16 centimeter level, if that means anything. These guys are particularly important because, or potentially important, they are obligate organohalide respirers. They need organic chlorine compounds to breathe, and they use hydrogen as their favorite energy source. So they could compete with antigens because hydrogen is another one of those things that could alternatively go to methane production. We did see this curious result just looking at some of our soil water data. In this one basin, chloride increased steeply in the first part of the growing season. We know that wasn't due to evaporation and concentration because the other things in the landscape stayed the same. They were all exposed to the same conditions. They're all pretty much lakes. It wasn't, so this is not all just evaporation, or else we'd see it here too. And looking through the metagenome, we see lots of chloroperoxidase enzymes. These are enzymes that can be forming organic chlorine compounds in the soil. A lot of different microbes involved in this. Others that are breaking down organic compounds with chlorine on them. And even got a little bit into understanding the nature of the organic chlorine in the soil. It increases with organic matter in general, just like you'd expect. The more organic matter there is in the soil, the more organic chlorine. But there are these two different slopes. So the ancient and older soils have the steeper slope. So as soil organic matter is accumulating, developing in these old and ancient basins, it's getting enriched in organic chlorine, which is kind of cool. And I had to show off a little bit of this Zane spectra. This is X-ray absorption near-edge spectroscopy for chlorine. And this shows that we have some plant samples like moss, we have sedge, grass. Those have very similar spectra to these young and medium soils, but the old and ancient ones start looking different. This is a principal component analysis where you can see all the vegetation samples are clustered with the young and medium soils. The old and ancient ones are all over the place. So plants might be an important initial source of this organic chlorine, but then it starts getting chewed up and reformulated in soils as they develop. So, there does seem to be an active biological chlorine cycle in the Arctic coastal plain, lots of microbes involved. Potentially, this could be another competitive pathway that would compete for hydrogen with antigens. Could have implications for ion cycling because those two cycles do interact. And I should mention, people have studied the fate of things like methyl halides because they're very important for organic chemistry, ozone-destroying reactions and things like that. But no one knows anything about the internal cycling of organic chlorine in the soil, so we think we have a niche to study this. And again, the main point of this was I didn't even know that this pathway existed about 5 years ago. I was just looking through my molecular data. Dehalo respiration? Wow. So I'm a microbial ecologist. I love the fact that given anything that's, if there's a pathway that's thermodynamically possible, some microbe out there will be doing it. And so this is just another example of that. So with that, I want to thank you all for listening. I want to thank Matt for inviting me and everyone for being such great hosts. And long list of students, collaborators, assistants, field support, etc.
The organochloride reduction, where is that on the delta G sequence?
Well, that's a really good question. That's really sort of one of our research questions. Now, as far as I can tell, I was expecting it. Let me go back to this tower way back then. Originally, based on some published work, I thought it was going to be somewhere here. I thought it was going to be there, above iron. By the way, this iron thing, of course, totally depends on what mineral it is.
Yeah.
This is drawn as one line, but in terms of this is like redox potential. If it's like chelated iron, it's up here, and if it's like, you know,
Yeah.
some other highly crystalline mineral, it's much further down. I just saw a paper where iron was interfering with this organohalide respiration in a pure culture, and where it seemed like they were preferentially doing iron reduction. So it made, that's why I drew it here, is I read a paper recently where it made it look like iron actually might be a preferred electron acceptor. But again, this is, chlorine and iron are two extremely diverse subsets, sets of compounds. So I wish I could answer your question better. This is my guess. Yeah.
Yeah, up here in the Arctic, kind of the top of the world, where you don't have these huge mountain ranges washing down to the system, how much iron is in the system, and what's the source of new iron?
Right, so there is some, there is dust. I always love, I don't know if you've seen the NASA animation of dust moving around the planet, the Saharan dust that moves around. That shows up all over the planet. The Brooks Range is, I don't know if I have, anyway, Brooks Range is there right around the Arctic Circle, and the Arctic coastal plains everything north of the Brooks Range. There are some rivers that come from the Brooks Range, and they carry sediment down to the Arctic Ocean. And that would deposit mineral material. But what I've assumed, and what seems to be the case for, based on just what I see across the landscape, that underlying marine silt that was there before the organic matter started developing seems to be the primary source of iron. I didn't actually mention that at all. That all these soils are built on some like marine silt sediments that are there, and then when the areas drain, they fill up with vegetation and become peatlands, peat bogs essentially. And so that mineral layer is showing you is, marine-derived silt material. But dust inputs, there's, again, I try to stay out of the realm of atmospheric people because I feel like I don't know enough and I'll get in trouble. So dust inputs are probably very important, and there are at least areas where rivers, that landscape, not only is it filled with lakes, but there's also all sorts of little streams and rivers coming out. So that material is being transported from the Brooks Range, ending up in that environment.
Does it need to be transported? Can it be abiotically oxidized through?
Oh yes, yeah, absolutely. No, it cycles, right? Absolutely, it does cycle.
Possibly,
Yeah, and I'm sure that it cycles more quickly than it's arriving by dust.
Yeah.
Because that has been the challenge, though. The simplest explanation for how the iron 3 gets regenerated is that over time, over the season, the water evaporates, the water table drops a little bit, and gets reoxidized. That's what would happen in typical wetlands. But it's a little bit non-intuitive from what we've seen. I didn't draw attention to it in the graph, but there was a phase of oxidation that happened in my seasonal time series that happened when the landscape was the wettest, with the high, and so there could be whole cascades of electron transport schemes where the iron is oxidized by something which is oxidized by something else, but the oxygen does it, but how does it get there? My favorite idea is the conductivity through the soil. People have found that soil can actually make conductive networks, so minerals, biofilms, humic acids, can actually conduct electricity, not the usual way of ions floating around a soil solution, but around the surface of these particles. So there's a cool study that came out not long ago where they have sulfide being oxidized in the sediment. They turn on and off the oxygen above it, and turn on and off the sulfide oxidation at a much faster time scale than you could account for by diffusion, showing that there was little nanowires, like that. So I'm hoping to find something like that in the Arctic. And then there's roots that could be transporting oxygen. Yeah.
So I have more of a question on your functional diversity of your microbial diversity you're looking at. It seems as as you guys, meaning microbial biologists, start to look in more detail and look at more sequences, you're finding a tremendous amount of diversity as your rarefaction curve showed. And then your comment was, basically, if there's a pathway out there, some microbe's going to be doing it.
Yeah.
Last week we saw Forrest was here, and he was talking similar, right? And for someone like me who's a field ecologist, I can't even imagine that diversity. When we start comparing terrestrial systems for macro diversity, you become comfortable with talking about tens of species to hundreds of species to thousands of species. Wow, that's tremendous changes in diversity. What's a tremendous change in microbial diversity that matters ecologically?
It matters ecologically.
I mean, I'm just looking at this going, everyone's talking about gazillions of sequences and OTUs that are in the thousands. Is it 3,000 to 30? Or is everything just so microbially diverse around the planet?
Right. We ask ourselves that question a lot. It's like, it seems so redundant, right? Like, there's no way you could come up with that many niches. So, I mean, it's interesting. Spatially, there's all sorts of, there's a lot of good models to explain why things are that diverse. But in terms of, I should correct myself. People have done some cool studies at linking diversity and function at the microbial level. And that was surprising that they found anything. Because with plants, it makes sense. You manipulate the diversity, you lose an important functional group with the nitrogen fixers, the productivity goes way down. Apparently, there have been a couple studies, and I haven't looked at them closely. They knock out the diversity with even things like chloroform, just like, okay, let's just beat the microbial community with a stick. Fumigate it, and half the species die, or 9/10 of the species die. And then look for changes in the functionality. So are there enzymes that actually disappear, or probably not disappear, but become a limiting factor? So apparently you can hit a microbial community hard enough to get some functional response. I'm used to thinking of them as so diverse that they're incredibly stable, so they're actually a good endpoint for looking at that whole diversity, stability thing. They're not very invadable systems, right? Like, they're usually, presumably, the microbial community, it's hard for one thing to come in and take over. Right? Are there invasive microbes? Is the other thing we always ask ourselves, because that would be cool. There's diseases. We know that diseases happen, but that's not happening because it's taking over the microbial community. It's generally just having a big impact on some other trophic ones.
So you've got your tundra system, you've got reefs, you've got the deep benthos. Can you, I guess if you flip it the other way, can you think of a system that is microbially depauperate?
Well, the first most tractable systems were this extreme, the acidophilic, the iron oxidizers and like Iron Mountain, where the pH gets to like -1, literally, where there's 10 molar sulfuric acid. That was a place where they were able to sequence the entire community years before, when the sequencing power was still less than it is today. And there were 4 species there, so that was pretty impressive. Like, that's really extreme. I just thought of another example where it's human guts are, I don't know if Forrest talk about human guts, or is he about corals, or he's all over the place. Anyway, human guts are pretty diverse, but not anything like soils. There's probably 800, say, things that look like species compared to 50,000 in the soil, in the ground soil. People who have chronic bowel disorders, various types of illnesses, they do seem to have lower diversity in their guts. And so again, I don't know if that's a causal, or which way the causality goes on that, but that's a system where there might be a strong diversity function relationship there. Anyway, but those are the kinds of questions I love to ask myself. I can only give you more questions.
As a follow-up.
Yeah.
I was just wondering about soils in the farmlands, like around here. I've heard speculation from landowners that they put plastic over it and then fumigate the soil to try and kill parasites in the soil. Do you think that would have an effect at a level that you would be able to detect?
I think so. And probably by now people people have done some good studies on this. The last one I looked at was kind of anecdotal because there wasn't a lot of, they were just like, hey, we have this power to sequence, deep sequencing of soils, and we can really start to look at diversity. And they included one agricultural soil and a few forest soils. So, and I'm sure there's better studies than this. This just happens to be the one I remember off the top of my head. And I couldn't help but noticing the agricultural soil was less diverse than the others. But that was sort of anecdotal. But yes, I mean, when you plow things, when you disturb soil, you're breaking up the aggregates. There's really cool studies showing soil, diversity of the microbial community in soils depends on, just like on a larger scale, spatial heterogeneity. So if there's more connectivity between pores, if you break up the aggregates, you know, you'll probably have a dominant competitor take over, where otherwise there were weaker competitors that were, were protected. So yeah, absolutely physical disturbance of soils. And I don't know what presumably chemical stuff too, but.
Would that translate into reduced production or anything like that? Soil health?
Well, I think the most obvious way are probably with plants, like the microbes that are interacting with plants, like mycorrhizal fungi. Now, for crops, of course, I don't know if we're talking about, for crops, they don't care about mycorrhizal fungi because they're just, none of those things are really mycorrhizal and they're giving them nutrients in it. But in general, for soil fertility, yes. Anything that's, the free-living bacteria that like fix nitrogen on plant roots, and there's a lot of cool work done on, this is an acronym I always stumble over, plant growth promoting rhizobacteria. It's a PGPRB. That's actually is a big interest in agriculture right now, and that is an area where having a relatively undisturbed microbial community in the soil, I can see, can even be good for agriculture. Although, if you have enough water and fertilizer, who cares?
I think I'm gonna let Dave off the hook, and thank you for coming.